All right, it's my pleasure to introduce Jeremy Schmit today as our speaker. Jeremy got his BA from Northwestern and then his PhD from UC Santa Barbara. After a postdoc at Brandeis and a postdoc at UCSF, he landed at Kansas State. So the transition from San Francisco to Manhattan must have been exciting.
Piece of cake.
And so he's been there since 2011. And he's going to talk today about some theoretical biophysics work that he does on amyloid aggregation, which hopefully some of you probably know is part of Alzheimer's. So let's welcome Jerry for talking today.
So thanks a lot for having me out, and thank you for that introduction. I'm going to talk to you about work I've been doing on protein aggregation. And before I do that, though, before I launch into the details and nitty-gritty, I want to start off with some big picture. And it doesn't really get any big picture than the central dogma of molecular biology. And all I want to do here is sort of relate the major classes of biomolecules and just sort of put them into context to each other. So what the central dogma says is the information, everything that the cell needs to know in order to do anything the cell might want to do, is encoded in the DNA. And so when the cell decides it wants to perform some function, what it's going to do is it's going to extract that information on the DNA. It's going to go through a couple of intermediate molecules, and that information is then going to be translated into a protein. And a protein is really where the rubber meets the road in biology. This is the molecule that performs all the functions in the cell, with the big exception, of course, being information encoding and the cell membrane, perhaps. But any sort of catalytic function, enzymatic function, signaling, structural, that stuff is all performed by the protein. So the proteins are your primary functional molecules. These are the things that do the stuff. Okay, so what is the protein? Protein is a polymer, which basically means it's a long stringy molecule built up of repeating units of the same thing. So down here, if you look at the backbone here, we have this nitrogen, carbon, carbon, nitrogen, carbon, carbon, repeating unit here. And there's two features of this chemistry that I want to point out to you. The first of all is this feature outlined in the box here. We've got this double bonded oxygen here and the hydrogen hanging off the nitrogen. And that box there actually encodes an electrostatic dipole. So this oxygen is slightly negative charged and the hydrogen is slightly positive charged. And so this is going to be important later in the talk. What you have here is a backbone that has a series of electrostatic dipoles that are oriented roughly perpendicular to the backbone of the polymer. The second feature is down here at the bottom here, this R group, R1, R2, R3. And these are what we call the side chains of the amino acids. And these are what distinguish one protein from the next. And these side chains can be a number of different things. Some of them are charged. They can be positive or negative. Some of them are hydrophobic. And still others are hydrophilic. So they'll want to stay solvent exposed, while these guys will want to try and hide from the solvent. So when the ribosome spits out a protein, what you have is this sequence of side chains, all of which are slightly different. And they all want to be in slightly different chemical environments. And so this leads to a frustrated situation where you've got all these groups, they all want to be in different states. And so what the molecule will do is it'll try and collapse into what we call a folded state that tries to satisfy as many of these different requirements as possible. So in this case here, what we have is the hydrophobic side groups that have packed themselves into the core of the protein in order to get away from the solvent. We've got some positive-negative pairs out here on the perimeter, and it's going to try and do this as best as it can in order to satisfy the requirements. And it's this folding action that gives the protein its function. These are the things that give the protein the shape it needs to perform its function, and the thing that's going to align the chemical groups into the correct geometry in order to perform whatever catalytic or enzymatic activity it needs to perform. So this is, of course, a very simplified physics type of view of it. When we look at real molecules, the picture is far more complicated.
What do you mean on frustration?
Meaning that each one of these different colored balls wants to be in a different state. Okay? And so it's going to try and satisfy as many of these requirements as possible, but in this case here it fails. Okay? So this is a hydrophobic thing that remains solvent exposed. and it's just the best that it can do. That's what I mean by it's a frustrated state. It can't satisfy everything at once. Okay, and so, you know, it's if something is exposed in a situation here where it's not satisfying its ideal wishes, most likely that's going to be something that's essential for performing its essential function. That's going to be the catalytic site or something like that. Okay, so we have very complicated viewpoints, and of course this is way more complicated than anything you're going to get any useful information out just staring at. And so we have various simpler representations. So this is just a view of the same molecule, leaving out all the side chain information and following the backbone as you go through it. Okay, so this process, this folding problem, is known as basically the signature problem of protein biophysics. And what is the folding problem? The folding problem is trying to understand how a molecule gets from this disordered string-like state into this functional compact state. And why do we want to know about this? Well, from the various genomic projects, the human genome and all the genomes from many other organisms that have been determined, in principle, we know what every single protein is in the human body or in any organism that's been sequenced. We have the complete sequence of amino acids there, and that should tell us everything we need to know about what that molecule is and what it does. But we don't know how to interpret that information yet. We don't know how to take that genomic data and get from this state to this state. And if we could, then what that would mean is that every time a medical researcher identifies this particular gene, for instance, as being associated with cancer, then we'd be able to take that gene, figure out what it is, and try and figure out how to design drugs in order to come back in and either activate it or deactivate it and do stuff with it. But we don't know how to do this step yet, and that's what we'd like to be able to get to. So the step in going from here to here was proposed by a biochemist known as Amyloid. And what he proposed is that the folded state here represents the thermodynamic ground state for a particular protein. So this here should be the state that has the lowest free energy of all the different states that this can adopt there. So that provides a recipe, if he's right about that, and we think that he's mostly right there, except in a few cases that actually will be the entire second half of the talk. But for the most part, this seems like a good working hypothesis. And so the question is, how do we go from some sequence information to find the lowest free energy state for a given molecule? And this ends up being a pretty difficult problem to solve. And to illustrate that, let's take a very typical protein. In fact, we're going to take a smallish protein, one that consists of only 200 amino acids. And let's just say, for just wild argument, let's say that each one of these amino acids can only adopt 3 different states. So this is, of course, a lowball estimate, but it'll get us started. So what that means is that, that this number of states that this molecule can adopt is something on the order of 3 to the 200. So if we assume that the system's ergodic and it's sampling all these different states, how long would it take for it to find the lowest free energy state? Well, a guess would be that the protein can sample a state every nanosecond, which is characteristic of a very small scale local rearrangement of the protein. It's not good enough for a large scale rearrangement, but again, we're making very conservative estimates here as to what this would take. So that means that this molecule is going to find its thermodynamic ground state in 3 times 10 to the 86 seconds. Or to convert that into units that might be more familiar, it's a fairly large number times the overall age of the universe. Now this problem is known as the Leventhal paradox, and basically what it says is that the search for the thermodynamic ground state cannot be a random process. Okay? That there's something that's helping the protein to get from its initial state to its final folded state, and of course a big part of that's got to be evolution. Evolution is not going to tolerate numbers like this, and so evolution is going to help the molecules to evolve in such a way that help it get to these low free energy states faster. In fact, actual folding rates are on the order of milliseconds to minutes. So these are very reasonable timescales for life, where this obviously is not. Okay, so how does a protein go from its initial state to its final state? Well, the best picture we have for that is something like this, in that the free energy landscape is something shaped like a funnel. And basically what this says is that we start from this disordered state here, and the protein will start to restrict its configuration space by making some local contacts that, again, this constricts its configuration space, and now some other contacts can form which constrict the space even more, and eventually we get down to the thermodynamic ground state. Of course, this is a very high-dimensional search space because all of these bonds have their own dimensions, so perhaps a more accurate way to represent this would be something like this, where we have this large dimensional search space up here, and of course we can form incorrect contacts too, which gives you a very rough free energy landscape. You're likely to get trapped in a local minimum over here, and you need a thermal fluctuation to kick you out before you can eventually find a pathway that gets you down to the thermodynamic ground state. Okay, so with this conceptual understanding, you would think that, okay, we should be able to solve this protein folding problem. And depending on how optimistic the person you run into, you can get different answers on how solved this problem is. My claim is that we're still pretty far away from being able to actually address this problem, and we're certainly nowhere near being able to take a gene that's associated with cancer and be able to diagnose what that molecule is and what it does. And so what are the problems here? Well, one problem is that if we are going to copy nature and we're going to allow the dynamics of the folding process to take us from the initial state to the final state, we have to be able to understand the forces that are interacting between the molecules. And again, my opinion is that we're a very long way from actually being able to understand these forces. What you have here is, in these molecular simulations that people use to fold proteins, you have this high-dimensional force field, which basically means you just have lots of different parameters that describe the forces between this type of atom and that type of atom. And this is a very highly underdetermined problem because there aren't nearly enough ab initio calculations and experimental data that you can do to uniquely constrain all those different parameters. And even worse, I think in a lot of cases, we're actually even using the wrong functional forms in these cases, and there's very little work into trying to figure out what functional forms we should be using. Another problem in the proteins is that there's no separation of length scales. If you're trying to determine overall features like the shape of a protein, which just ought to be the simplest thing you can get at, is what is this protein shaped like? Is it round? Is it egg-like? Is it sort of rod-like? This is determined by very short length scale perturbations in the sequence. These small angstrom scale side chains are what determine many nanometer long features in your final folded protein. So when you're trying to figure out these protein characteristics, you need all the little microscopic details in order to understand even the biggest, most grossest features. And so that leads you to a problem in timescales and computational power. Because here's what you're trying to do. You're trying to understand protein folding events that are happening out here on the milliseconds to seconds timescale. And this kinetic process is determined by angstrom scale chemical perturbations in your sequence. So what that means is you care about things like bond vibrations happening on femtoseconds. So this sets the elementary time step in your computer simulation. Okay, 1,000 times slower than that, you see things like water relaxation happen. Water relaxation is important for things like the hydrophobic effect, which is one of the major driving forces for this polymer collapse. And then another 1,000 times slower than that, you finally start to see the backbone of the protein rearrange on the length scales necessary to start to see some conformational rearrangements. This is where you start to see those backbone dipoles form the hydrogen bonds that lead to alpha helices and beta sheets that allow them to desolvate and form these collapsed structures. Okay, so you go through 1 million computational time steps here before you even start to see the protein start to relax on any time scale that's significant for that molecular rearrangement. And so when you put these requirements into the fastest computers available on the planet, what you find is that your molecular simulation timescales peter out at around the microsecond timescale. With a few, studies that are maybe able to push out to milliseconds, but those are exceptional because as soon as you push out, it's kind of an automatic science article before you even do science on it. Oh, that's not true. Okay, that's disparaging. Okay. Okay, so this is the problem in protein folding. Okay, I'm not interested in protein folding. I'm interested in aggregation timescales. And so what kind of timescales are you interested in? Well, we're interested in things like Alzheimer's disease. And Alzheimer's disease, if the timescales you're interested in, of course, the onset of disease, the progression of disease, this isn't measured in seconds. This is measured in decades to even centuries. Okay, so we've got a problem here. This is another deal altogether. And so this is really where I see my role coming in as somebody that does biophysics theory, is trying to understand what's going on in this, call it 21 order of magnitude separations between your molecular timescales and your disease timescales. Okay, so let's back up. Let's talk about protein aggregation in very general terms. Okay, amyloids are aggregates that show up with many, many diseases. There's lots of good evidence to believe that these aggregates themselves are causing the disease. I'm not going to get into that right now. But the list of diseases is a very partial list is over here. Alzheimer's, of course, is the headliner. And what you see is if you do an autopsy on somebody with Alzheimer's, is you see these extracellular deposits of insoluble protein, these little clumps, these masses here. And something associated with these clumps, these protein clumps, leads to neurodegeneration, cell death, eventually dementia, and all your classic Alzheimer's symptoms. So if you look at these aggregates, or you try and reconstitute them in a lab, what you see is that the proteins have joined together and formed these long filamentous aggregates, these essentially one-dimensional, unbranched, but often bundled together aggregates. And these they clump together and form these insoluble masses. So what are they? If we zoom in on them in molecular detail, what we see is that the floppy molecules, what they've done is they've all lined up straight with each other and basically stacked on top of each other like this. And what that allows them to do is that allows them to line up those electrostatic dipoles on their backbone so that they can form hydrogen bonds, which are shown by these red lines here. And so those of you familiar with biology, this is a conformation known as the beta sheet. And so an amyloid fibril is essentially just an infinitely long beta sheet. So the fibril axis in this case here is extending up and down from the floor to the ceiling. If we were to look down this beta sheet, down the fibril axis, it would look something like this. And what we know is experimentally is that these single beta sheets, these isolated beta sheets, are probably not stable in solution. And so what you need in order to get a stable aggregate is you need a couple of these beta sheets to laminate on top of each other. And what that allows them to do is the side chains from the proteins will then interdigitate with each other, and that provides some additional binding energy that helps to stabilize the aggregates. So this is the cartoonish view of what's going on. If we look at the molecular picture, this is the structure of an A beta aggregate. This is the molecule causing Alzheimer's disease. And what you can see is that each molecule here is folded in half. So you have a blue piece and then a green linker and then the red piece here. So this would be the blue, the green linker, and the red piece. And so each molecule forms two of these beta sheets, 1, 2, and then you've got two molecules, 3, 4, and so it's 4 beta sheets thick. Now the other point I want to illustrate here is that these aggregates are incredibly highly ordered. So if we look, each color here denotes a different type of chemical group, and what you'll see here is that all the purples are all stacked right on top of each other. All the blues are stacked right on top of each other. These are little 6-membered carbon rings. They're all stacked up right on top of each other. And so basically these molecules resemble one-dimensional crystals. So everything is not just stacked on top of each other, but it's perfectly aligned so that all the side chains are lined up in perfect registry with each other. Okay, so I'm over and over again going to start coming back to timescales on this. And so I've already mentioned that the disease timescale that we're interested in is this thing out in decades to centuries. that describes the onset and progression of something like Alzheimer's disease. And this is, of course, is a fairly inconvenient timescale, not just computationally, but experimentally, because there aren't too many graduate students that want to stick around long enough to do that experiment. So what you do, or what every laboratory studying this aggregation process in the laboratory does, is try and find conditions where you can accelerate this aggregation process up to something that's experimentally tractable. So what you do is you work at extremely elevated conditions, or you use solvent conditions, things like elevated temperatures or denaturant molecules, anything you can do that'll accelerate this process and get it to happen on a time scale where you can do lots of experiments, where you can establish reproducibility and collect data. So the problem is that there's no unique way to do this. As I say, you can go to extreme concentration, you can go to extreme pH, extreme temperature. There's lots of different recipes you can do in order to make this go faster. And not so surprisingly, when you do that in lots of different ways, you see lots of different things that can happen. And so the literature is filled with these apparently contradictory results of all the different things that can happen on these laboratory timescales. And so which ones of these, if any, are relevant at all toward the processes that we care about? So approaching this like a condensed matter physicist, what I would say is that what you're doing here is you're essentially looking at a high-dimensional phase diagram. And so depending on how you prepare your experimental conditions, these experimental groups are essentially just throwing darts at random at this phase diagram and seeing all these different states show up. And so what I've done in collaboration with this group down at UCSF is we've actually mapped out some of those phase boundaries. And so the punchline of this, I'm not going to get into the story in detail, but the punchline of this work here is that these aren't really true thermodynamic phase boundaries. These are, in fact, kinetic phase boundaries. And so the state you see at the end of this aggregation process is not so much determined by which state is more stable, but which one forms fastest under a given conditions. And so the role of the theorist in this case here is to understand the physics underlying these kinetic processes here so that we can extrapolate these phase boundaries out to physiological conditions and understands what's happening under these much slower, much more dilute properties going on inside somebody's brain.
Can I comment?
Yeah.
You've now just turned a corner and denied the hypothesis of free energy driving.
Absolutely, yeah.
And the free energy proposal had no time dependence in it because thermodynamics doesn't deal with that. And
So,
kinetics does.
okay, so that's a good point.
Why do people keep repeating the thermodynamic free energy thing unless it's just a straw man set up for colloquium?
It's not a straw man setup for the colloquium. It's sort of the underlying.
Is it your particular angle to attack it?
Well, okay, so just from the aggregation point of view, this fibril state over here, we have every reason to believe is the actual thermodynamic ground state. Okay? And it's more stable even than the single folded protein. That seems pretty clear. And until something else shows up that's even more stable than this, we believe that this is the thermodynamic ground state. So what you could say is that the folded state is possibly the single body thermodynamic ground state. And so if you put that one qualifier in, then it sort of restores Anfinsen's hypothesis as far as we know. Although in biology, if you can imagine a counterexample, it exists. And I think there are cases where you can have proteins that will fold into incorrect states that might be very stable that aren't pathological. But for the most part, that's your starting assumption until proven otherwise. And there will be counterexamples. In this case here, this is the thermodynamic ground state, and these here are higher energy states that are kinetically favored once you cross these boundaries. Okay, so one role of a theorist in this case is, of course, to establish these phase boundaries here. But there's one other place that actually I'm going to spend most of my time in this talk talking about. And to show you that, I'm going to go back to my timescale plot here. And again, once again, I'm going to put in the molecular simulations. So one role for a theorist is to understand this gap right here, But the other place is we have another timescale gap showing up over here. What is going on in this area here that we can't access with a high-resolution molecular simulation? Why is this happening very slowly? And in fact, that ends up being a pretty good question. Because naively, you would look at this and say these aggregates are nothing more than beta sheets. They're just secondary structure. Okay, these things are just stacking together and they're forming hydrogen bonds. And they do that in any kind of protein folding process. And we know that the time scale for the secondary structure formation is on the order of nanoseconds. So naively, you would expect that this aggregation process should happen in nanoseconds when in fact, even under the fastest experimental conditions, it takes about a second for each molecule to add to the aggregate. What on earth is happening here? Why is this so darn slow? And why, in particular, can't our molecular simulations see what's going on there? So the hypothesis I want to throw out here is that what's going on is that the fibrils actually suffer from this Leventhal paradox, this search over many different conformational states. So recall that in order for a functional protein to fold to its native state, what we need is some sort of free energy pathway that helps to guide it down and to restrict its conformational space. And this pathway shows up because billions of years of evolution have essentially excluded molecules that don't provide such a pathway. They aren't biologically useful, and so they evolve to something that is. Well, here we're looking at not a functional folding process, but a pathological aggregate. So there is no evolutionary pressure that's going to give you a nice pathway like this. In fact, if anything, evolution is going to be working in the other direction to try and remove such pathways. So instead of a nice funnel like this that guides us to the final state, what this would say is that we're essentially searching over a flat landscape here. And these molecules have to just diffuse around here before they finally get lucky and drop into this deep free energy well in the middle. So your aggregation timescale is essentially set by how long it takes for a molecule to explore this conformational space before it finds this free energy well illustrated here in the middle. So how long would that take to happen? So here's my cartoonish view of what's going on. So here's an established fibril, and I've got a soluble molecule coming down, and it's forming a hydrogen bond, and this molecule eventually is going to link up with it. Now there's a lot of different ways that this first molecule can link up. So I can make a first contact between this red and this black, or this red and this purple. And in fact, with these sort of 4 residue long molecules, there's 16 different ways I can form that first contact. But now recall that our final aggregated state is these very highly in register lined up configurations. And so there's only 4 out of these 16 that are consistent with that highly ordered state. So the question is how long does it take me to explore all these different binding states before I eventually lock into one of these ones along the diagonal here that's going to give me an ordered aggregate? So in order to solve that problem, again, here's my cartoonish view of what's going on. I have a soluble molecule that's going to come down. It's going to start forming these backbone hydrogen bonds. And what I'm going to do is I'm going to take this process of the formation and breakage of hydrogen bonds, and I'm going to map it to a one-dimensional random walk. So here's my random walker here. It tells me how many bonds I have. If this guy gets all the way over to the left, I unbind and float away. If he gets all the way over here to the right, that means it latches on and becomes part of the growing aggregate. So this isn't a completely random walk because these bonds are energetically favorable. So what that means is that this is actually a biased random walk. The walker would rather hop to the right than to the left. And then we can use detailed balance to relate those right and left rates to the free energy of these bonds. And that free energy works out that it's about 1.5 times more likely to hop to the right than it is to the left. Okay, so what we can do is we can take this information and we can solve this one-dimensional diffusion problem exactly in order to figure out how long the molecule is going to take in each one of these states. And in the model we're doing here, I'm not going to distinguish between what these colors mean. I'm just going to use an average binding energy for all of these states. And so what comes out of the model? what you see here is that if the molecule comes down and I get lucky and I bind in register where everything is lined up, this molecule will become part of the final aggregate very fast. Something on the order of nanoseconds, which was exactly our naive expectation for how long it would take a molecule to come in and bind if it's just forming the secondary structure. So this is in agreement with the back of the envelope guess. It's very fast, but it's not a major contributor to the aggregation time. What is the molecules that come down and bind incorrectly. This molecule is out of register. It's not blue is matched up with red, black is matched up with purple. This is unbound, but it doesn't know that. It's still forming these favorable hydrogen bonds, but it's not lined up in a way that's compatible with the further growth of the aggregate. So we spend a long time waiting for this molecule to break all these bonds and float away so that we can try again. How long does that take? It takes something on the order of a millisecond to happen. Much, much, much slower. Okay? Now, of course, if we think back to the previous slide, there were a lot more off-diagonal elements than were on-diagonal elements. So what that means is that we have to get it wrong roughly 10 to the 2 times before we finally get it right. And what that means, if we multiply this factor of 80 times a millisecond, what that says is it should take about 1/10 of a molecule before I explore this binding space and bind correctly and latch on and become part of the aggregate. So in looking for something that was 9 orders of magnitude, what the calculation eventually spat out was 8 orders of magnitude, which at the resolution of this modeling here I think is pretty reasonable. So what comes out of that? Well, with the understanding that this stuff here is very fast and this stuff is very slow, you can greatly simplify the aggregation expression, which I didn't show you before, and it takes a relatively simple form. In fact, this is exactly the form you would guess if you said I have a process that can be either diffusion limited or reaction limited. So here's a diffusion time, here's a reaction time, no surprises there. But the thing that is, there are 2 surprises I claim that come out from doing the calculation. The first of these is that you have a very small prefactor showing up in this calculation. And what that comes from is the fact that I have to sample these incorrect states very many times before I finally get lucky and one latches incorrect. In order to say that a different way is if you came along and told me the 2 fundamental timescales showing up in this expression, your growth rate expression would still be off by 2 orders of magnitude. The other thing that's interesting is that the reaction time that shows up in this calculation here is an off-pathway event. It's not the integration of a correct molecule coming in and becoming part of the aggregate. You're spending all of your time in this growth process waiting for this incorrectly bound stuff to get the heck out of the way. And so what's the prediction there? The prediction there is if you want the aggregation to proceed faster, what you want to do is you want this misregistered stuff to get out of the way faster. And so what you do is you actually weaken the bonds. And you can do that either by increasing the temperature, this is a 1/temperature temperature scale, or by adding denaturants, which are molecules that weaken protein-protein bonds. And in both cases, you see that the aggregation rate goes up as you weaken the bonds. You get this inverse effect. Yeah?
What's the L stand for in the prefactor?
L is the length of the molecule. Okay, so a typical length for one of these pathological molecules is about 40. And you throw in a little, you've got 40 different ways it can line up like this, but then it can also flip around and there's another 40 ways there, and that's where you get that factor of 80. Yeah. Okay. So the story here is that we're basically randomly searching over this configuration space before our aggregate can grow. And so anything we can do that will allow us to explore this space more quickly will help us grow faster, and we can smooth the gears here by weakening all these bonds so we can diffuse over the surface more quickly before we fall into the final state. Okay, so with that kind of rough understanding of what's going on in the growth process, let's move down in timescales even more, and let's try and attack something that's happened even slower, and that is the nucleation of the fibrils themselves. Okay, so growth rate happens on the order of seconds. The nucleation time scale is, hours or days. Okay, and so what you see is that if I prepare a solution of these aggregated or of these amyloidogenic molecules, for a long time they do nothing. And then at some point, you get a stochastic fluctuation where the aggregation starts, it takes off very quickly, and then it saturates. And how long you have to wait before that initial onset depends on the concentration you use. At low concentrations, you might wait, that's about 3 days here under these particular conditions. At higher concentrations, you might just be waiting a couple hours. Okay? But in all cases, there's this waiting time where nothing at all happens before you have to wait for the aggregation to start. So this is this is exactly what we see in a lot of different phase transitions. We call it nucleation-based behavior. And the classical story for nucleation behavior is that when you're starting to grow an aggregate at small sizes, the free energy of your cluster is dominated by the surface tension, which is repulsive. And so you actually have to climb uphill in free energy space until you hit a critical size at which the favorable bulk interactions now become large enough in number that they can dominate the unfavorable surface tension, and you start to move downhill. And so this waiting time before the onset of aggregation is essentially the time that your system sits down here in this free energy well over here, rattling around before there's a free energy fluctuation large enough to kick you up to the top of the barrier so that you can start going downhill on the other side. And so this is a process that happens in lots of different phase transitions. And in fact, many of you probably have it going on in your kitchen right now. So many of you have seen you buy a jar of honey from the store, and it shows up, and it's this nice golden brown liquid. You pour it out on your sandwich or whatever, and it sits there in your pantry for a few months, and then one day you pull it out and look, and you see this cloudy stuff showing up in there. And your honey's not going bad. It's not spoiled or anything. What's happened is that the sugar molecules in there have discovered that they can find a lower free energy state if they start to crystallize. And once this process starts, it'll quickly proceed, and in many cases, it'll fill up your entire jar. And what you find is that now your honey is not as nice golden brown liquid, but it's this sort of clumpy, crunchy, crystalline type stuff. And it's still perfectly fine to eat. But if you prefer to have this nice golden brown fluid that flows nicely, what you can do is you can take your honey jar and you can put it in a pot of hot water and very slowly heat it up. And if you're careful, you can melt all the crystal states in there. You can eliminate all the nuclei. And then once you put it back in your pantry, it'll stay stable there for several more months, and everything's fine. Well, mostly fine. There are some problems with putting your honey jar in a pot of hot water over and over again. The plastic doesn't like it. Okay, so where does this leave us for amyloid systems? Well, recall that these protein aggregates are actually these one-dimensional chains. And the problem is that when you're building a one-dimensional aggregate, your surface area doesn't change. Okay, so if I take the one-dimensional aggregate here, I add a molecule to it, there we go. Okay, I've gone from 1 unit of surface area here to 1 unit of surface area there. So my surface area hasn't changed here, and so my free energy as a function of my cluster size is monotonically downhill. So classically, one-dimensional aggregates do not have nucleation-based behavior. But that's clearly not the case in these protein aggregates. They're very clearly nucleation-limited dynamics. So what's going on there? Well, the answer almost certainly comes from the fact that they're not true one-dimensional objects. If we go back to our molecular structures of it, you can see that they've got a thickness. There's multiple molecules in this direction. Here, there's 2, and here you could even say there's either 2 molecules or 4 different sheets, and they have a width as well. And so this hasn't escaped the attention of previous authors. There's a bunch of papers using what I call matchstick models that basically treat the molecules as these rigid sticks that will then come together, and they form, and you get a fibril extension in this direction and some thickness in this direction. And now what you're doing is you're assembling a 2-dimensional object, not a 1-dimensional object. And this allows you to have a size dimension surface tension, which will give you a nucleation barrier. And as I said, there's a bunch of papers that have taken this sort of approach. Now, what I'd argue here is that I agree with them. This is absolutely true, but I think they're missing about half the story here. And the other half of the story comes to the fact that there's a free energy cost associated with straightening these molecules out and getting them to form these extended states here. And so what I want to do is I want to solve a very simple mathematical model that'll give us the dynamics of this straightening process and show how it will contribute to the nucleation behavior. And so that simple model is simply the nucleation, quote unquote, of a 3-molecule system here. So a very simple toy model here. So what we have here is we're forming a trimer here. And now what I need to do is I need 2 different reaction coordinates. Instead of that one little ball hopping left and right here, I need 2 reaction coordinates. One is going to describe the number of hydrogen bonds between these 2 molecules, which is 4. And one that describes the number of hydrogen bonds between these 2 molecules, 2. Now, of course, the system is completely symmetric, but my claim to you is that these hydrogen bonds are not the same. Okay? So for instance, let's look at this top molecule here. If I create another hydrogen bond here, if I bring this blue ball down and link it up there, I gain 1 bond in favorable energy, but I lose a unit of conformational entropy due to the fact that this bond is no longer free to rotate. So if this is a stable aggregate, I need the energy to win. I need this bond to win over the entropy, and so this is a favorable interaction here. Now let's come over here and look at this one. Let's say I try and form this bond right here. Well, once again, I get 1 bond worth of favorable attraction, but now it's going to cost me 2 units of conformational entropy because I'm freezing this bond and this bond. They're no longer allowed to rotate. So what's my free energy balance there? I've got 1 favorable bond plus a loss of 2 entropy. And in this case, the entropy wins. And this is an unfavorable interaction here. Okay, so we've got 2 reaction coordinates. We've got some favorable bonds and we've got some unfavorable bonds. And so once again, what I want to do is I want to solve this diffusion problem. And so, oops, that slide was meant to be deleted. Okay, and so here's what it looks like here. Okay, here's our 2-dimensional space. Here's my x coordinate. Here's my y coordinate. And I'm solving this convection diffusion equation where I've got these anisotropic velocities, drift velocities. And the drift velocities here, they're schematically illustrated in the arrows here. And what they say is that these bonds are favorable and these ones are unfavorable. And what they tend to do is they tend to drive me toward the diagonal where x and y are equal. So for every unfavorable bond, I've got a favorable bond in order to balance it out. So that should be intuitively reasonable. And so when you solve the dynamics of this diffusion problem here, and there's a lot of details that go into this model that I'm glossing over here, something interesting comes out. And that is, that's illustrated here. So what you see is this orange surface here. This is the free energy landscape here. And so, as you can see, there's this trough running down the middle here. This is where you have 1 favorable bond for every unfavorable bond. And so, the more of those you get, this is your fully aggregated state down here. And so, this is your lowest free energy pathway. Now, if you were to do, say, a saddle point approximation, you would say that what the molecule is going to try and do is it's going to take this lowest free energy pathway right down the middle of this trough here. But what we find when we solve that diffusion problem is that that's not what happens. Instead, what happens is these molecules prefer to climb uphill a bit before then attacking down and going to the trough from the sides. So instead of going over the saddle point, which is right here along the edge, they prefer to climb up the side ridges and then attack from the sides. Now, why is that? Well, it's not too hard to understand because this is the configuration sitting in our saddle point. This is the molecule that's got, or trimer, that's held together with 1 bond connecting each of the 2 side molecules with the central molecule. And a quick inspection of this says that can't be very stable. Okay, these 2 bonds are very weak. They're on the order of a half a kT each. And so you expect this molecule is going to fall apart very rapidly. Maybe I form a bond over here, but by that time the top molecule's fallen off. So this has a very, very low chance of succeeding. At the other end of things, you might imagine that this is a very high chance of succeeding. Say if these 2 molecules completely bind together and then a 3rd molecule comes down and attaches, this guy gets to form lots of favorable bonds very quickly, and so this has a very high chance of success. The problem is that this starting state here, where these 2 molecules came together and zipped up, is incredibly energetically unfavorable. Okay, this costs me a lot of entropy to do this, and so this is thermodynamically very rare. And so your best chance of nucleation success is somewhere in between, where I've got a little bit of an unfavorable nucleus to start from, And then the third molecule can come down, and it has a chance to form a few favorable bonds before things fall apart on you. And so it's these intermediate structures here, these sideways attacks that both avoid the saddle point and keep you from having to march too far uphill before you make your initial dive down into the valley. Okay, so this is just a simple trimer toy model, and I claim it yields some interesting physics. The problem is that the dynamics of it, when you calculate it, the nucleation rates aren't so good. And by not so good, I mean they're off by orders of Avogadro's number. And of course the problem there is that it's not just a trimer that's nucleating. You need to build a much bigger structure. And so we've done that. We've built a much more complicated model. I'm going to skip over this in the interest of time, but other than just to say that, that this model allows it to grow much bigger. Actually, we get about 10 mers before you get a stable nucleus, and the nucleation rates are, order of magnitude comparable to what we see experimentally. What I want to spend the last few minutes talking about is how we've taken these insights from the analytic theory and tried to use them to put the details back in. Again, the thing that discriminates one protein from another is the fact that you've got these very high resolution details encoded in the amino acid side chains. And this is what you ultimately care about when you're talking about diseases. Why does this particular family get Alzheimer's disease when this family is no more likely than the rest of the community? Well, it comes down to these small details. So what's the theory telling us about how we can bridge this big gap between what the computers can do now and what we'd like to be able to do? Well, what I'm going to do here is borrow from the state of the art in the protein folding community. And what they're doing is they're building what are called Markov state models. And what they do is they use long unbiased simulations in order to identify the local basins in free energy space. And so what they do is they look at these simulations and say, okay, we see a bunch of molecules that are in states very similar to this, and this, and this, and this, and they identify where they see clusters of states that repeat over and over again. Then they go back in and use some advanced sampling algorithms in order to measure the conversion rates between, say, these two states, and these two states, and these two states. And once you understand the rates of interconversion between these states, now you can put together large ensembles of trajectories that will take you from your starting point to your final point using all these different pathways that might connect you in between. Okay, so this is what we'd like to do in understanding these aggregate structures. And what the theory tells us is that this is actually much simpler to do than it is in the protein folding world, because our diagram of states consists of these linear chains of just forming and breaking hydrogen bonds, and then taking these molecules and shifting them by one molecule in registry to the left and to the right. And so what we've been doing in collaboration with Jinhan Chen's group at K-State is using lots of very short parallel simulations in order to sample these on and off rates for each and every one of these hydrogen bonds in a sequence-specific manner. And so then that allows us to build large ensembles of trajectories that take us from this state, sampling all these various states, and finally getting us to the aggregated state. So here's what it looks like. Over here. Okay, so this is the all-atom simulation right here. We've got an established fibril template here, and we're waiting to see how long it takes this new molecule to add on. And what you can see is it's fluttering around, and it's going to come down, it's going to form some bonds, it's going to break some bonds, it forms and breaks the bonds. And we run these simulations long enough that we can see a bunch of these bond formation and breakage events. We measure how long it takes, we fit that to an exponential, and then we've got the formation and breakage rates for all these different bonds. Now we come along and we build a number of larger trajectories of these events, where we can sample these molecules coming in and trying out all these different binding alignments and binding states. And so it comes in, it's going to form a few bonds, it breaks off, it forms a few bonds, and it goes through this over and over again until finally it'll happen to latch in with the correct registry and fully bind and eventually become integrated within the aggregate. So this stationary one here represents the whole fibril, which is not shown. Okay, so what have we been able to get out of this? Well, This is still really new, really green work here, so everything here should be taken with a bit of a grain of salt here. But we claim that we're actually starting to get mutation-specific growth rates out of here. So this blue molecule here represents the aggregate growth rate of a particular sequence fragment of the A beta molecule. And what you can see here is that if experimentally, well, these are simulation data, but these trends here are qualitatively consistent with experiments. If we do 2 different single mutants on it, this aggregation rate grows faster. That's the green line and the red line. So these are both single mutants of this, but if you make both of those mutations at the same time, what happens is you get a growth rate that's not as good. So what happens here is that some is good, but more is not better. And again, this is qualitatively consistent with the experiments. The experiments see that both of these single mutations help you, but if you make both of them at the same time, you don't get an additive effect. And so now what we're doing is we're going through this ensemble of trajectories and trying to figure out what it is about this double mutant that's actually slowing down the overall aggregation rate. And so with that, I will put up a summary. In my opinion, the interesting thing about these amyloid diseases is that you have this incredible range of timescales that you're interested in. All the way from nanoseconds describing your molecular dynamics, all the way up to decades and centuries describing your disease timescales. And this is a tough sell with the biological audience, but right here I can say with a bit more confidence that analytic theory provides a real nice tool that can come in and bridge this huge range of timescales. In fact, I'm not sure there's really any other tools out there that can cover that range. Those theories that can come out of that can also provide you good insights for putting in the high-resolution models in order to see what really is going on in atomistic details, which is, of course, the big weakness of an analytic theory. And in this case, what I've showed you is that these intermolecular hydrogen bonds provide a good reaction coordinate in order to make that, to make that jump. With that, I'd like to do some acknowledgements. This is my current group here. The nucleation calculations were done in collaboration with Larry Zhang, and Jinhan's group at K-State did all the molecular simulations that I showed you at the very end. And of course, I want to thank the funding agencies, and I'd be happy to take any questions.
Questions?
Can you go back? What mutations were you using?
Okay, so okay. So this is what is it? 16 to 22 of amyloid beta, and the mutations that we're using the mutations that the experimentalists used, and from the looks of this, these are non-natural amino acids. These are cyclohexane side chains that they plugged in. I think they were replacing phenylalanine, and so they were trying to find something. They were probing a range of hydrophobic, and so they were trying everything from alanine to cyclohexane, and see these are the very hydrophobic side chains that they were using. Yeah.
Let me see if I understand this idea that you have about the time scale that you came up in your first slide.
Uh-huh.
The time scale that we actually see experiments. When you take this huge molecule, right, very long molecule, and to calculate to begin with, you just say, okay, everything that happens at random will happen with the same kind of probability, and so let's calculate how many ways you can fold this. But actually, I would assume that these molecules just see their friends. The atoms just see their next door neighbors, or something, friends, or maybe friends of friends. That's about it.
Yeah.
So to go and talk about 3 to the 200 is not really, well, obviously it's wrong, but
right.
But even if you think about it without knowing anything, that's not the kind of number that you would come up with, right? Because, so it seems to me, and that's what I'm trying to understand.
Yeah.
It seems to me like in the end you show...
But there's also some very definite non-local contributions as well. I mean, when these things, so there's a lot of local stuff happening too. That's your secondary structure. But then you have the tertiary structure, where these, say, two helices will come into contact from very far away. And so there's
Right,
a
later
mixture.
on, First you start folding locally.
That's right.
Then slowly, so it's kind of a hierarchical problem.
That's right.
It seems to me like when you talked about this parallelization of the code and this picture, nice picture that you showed in the end, this is actually what's happening. I mean, everything is happening at the same time in different parts of the molecule, and that's the way it folds, not that you try all kinds of configurations to come. I mean, things are much more local than what it seems, and that's what I understood from this last kind of figures.
I think that's a fair representation. Yeah.
So you mentioned specifically molecular dynamics style simulations, but there are a whole range of other more coarse-grained simulations. I assume you didn't have luck with things like elastic normal mode simulations and things like that. Is there some particular reasons that those don't fit well with the new add-in type model?
Well, the problem with those simulations, there's no question that those can contribute a lot. The risk for those types of simulations is that you get out of them what you put into it. And so, there's been a lot of attempts to simulate, these sort of amyloid type structures. And so what they'll do is they'll use a coarse-grained model that, is hydrophobic on this surface and has hydrogen bonds on this surface. And they go, hey, look, we formed aggregates that form hydrogen bonds and hydrophobic surfaces. And okay, you got out of it what you put into it, and there's no real way of connecting those simulations to experiment and doing, a real comparison here. Now, this is even lower resolution still, but what the thing that allows me to do is actually go in and compare to experiments and do a bit of a sanity check and make sure that I'm on the right track. And I glossed over a lot of that except for the temperature and denaturant studies. There's just not much in the way of nucleation data, so I can't do much there, but the phase diagrams I can also do that for.
Sorry, I guess I was thinking a little bit more on like after you have your very coarse-grained model, the next addition step. But I guess since you already have access to a collaborator with full all-atom capacity, and that seems to be working. I was just curious, have you looked at?
No, in fact, if anything, we're going to go the other way. The next thing, we're using implicit solvent for these simulations, and we're going to go. It's working well enough, we're going to go straight to explicit solvent. And the nice thing is we think we know how to compartmentalize everything tight enough that, it doesn't do us any good to coarse-grain stuff out. Let's just throw in as many details as we can, and, we won't be able to run them very long, but we don't need to with this formalism. We can keep everything very short and parallel.
I was wondering if there were any other species of this phenomenon like Alzheimer's that we see. Or is it not?
Yeah, so there's mouse models for Alzheimer's disease, meaning we've cloned in genes into mice that cause them to get Alzheimer's disease. And so this is, one of the things that lets us know that the proteins themselves, that these proteins are actually causing it. Because if we introduce it into the molecules or into the organism and overexpress it, then they get Alzheimer's disease or something very similar to it. In terms of, of course, prion diseases, of course, also fall in this broad category of diseases, and so you have things like scrapie and mad cow that are very similar. So yeah, I would say the answer to that question is I don't know if there's an exact analog to Alzheimer's disease other than the mouse models, but there probably is.
What was the question that the answer is yes to? We couldn't hear it.
The question is are there any other organisms that have Alzheimer's disease? And so we can artificially create Alzheimer's disease in the mice by introducing these proteins into them and overexpressing them. And we can, prion diseases, mad cow and scrapie, show up in cows and sheep respectively. And these are, it's not Alzheimer's disease, but it's very similar type of aggregation disease. And so, I'm not sure if it's exactly Alzheimer's disease, but yeah, you see very similar stuff happening in other organisms as well.
So if we know the specific protein how important is it to typical brain function? I mean, certainly we could just develop a drug that blocks the protein or something, right?
Right.
So, yeah.
Okay, so this is actually a fairly small fragment of a protein. What causes it is you get this giant protein, and the part that causes the problems is just a small tail of it that gets cut off. And so many of the trial treatments for Alzheimer's disease that have been introduced to the market so far have been to try and block that cutting off. Like, let's just keep it from cutting off that tail, and if we can keep it bound to its big mother protein, then you won't have problems. They haven't worked terribly well, and I don't know why. Yeah.
Are you able to get medical results from your in vitro aggregation?
What's that?
If you're speeding up the aggregation, in
Yeah.
the lab, are there things you can tell about the actual onset of the disease from that?
Well, I mean, nobody really knows how to interpret any of those in vitro experiments because they are so much faster and at such grossly, in my opinion, such grossly unrealistic conditions that nobody really knows what they mean in vivo. And that's, that's part of what I'm trying to do here is help people decide which ones are relevant and, which ones might be representative of what's happening in vivo and which ones are not, by providing the functional forms people need to make that extrapolation.
No other questions? Let's thank our speaker again.
Thank you.